Cardiovascular disease is driven by a paradox. The very biological mechanisms designed to repair blood vessels end up destroying them. Oxidative stress and systemic inflammation are locked in a continuous feedback loop inside the arterial wall, turning localized tissue damage into metabolic syndrome and progressive vascular decline. To break that loop, we have to look past macro-level symptoms and focus on molecular chemistry. Specifically, the structure of a highly specialized xanthophyll carotenoid known as astaxanthin. While it shares a long, non-polar carbon chain with molecules like beta-carotene, astaxanthin is chemically distinct. It features two highly oxygenated groups, a hydroxyl and a carbonyl group, attached to each of its terminal ionone rings. This specific chemical architecture gives astaxanthin both highly polar ends and a nonpolar center. Because of this exact geometry, it aligns perfectly across the lipid bilayer of human cells. The polar rings anchor into the aqueous surfaces, while the carbon chain spans the hydrophobic core. This transmembrane geometry is what allows astaxanthin to intercept the biological progression of atherogenesis. Because it spans the entire cell membrane, it can interact with multiple biochemical targets simultaneously. It operates across three distinct biological nodes, acting as an intracellular and extracellular radical scavenger, acting as a suppressor of genetic inflammatory signals, and a mechanical enhancer of reverse cholesterol transport. Astaxanthin serves as a precise structural intervention tool, inserting itself into the cell membrane to dictate how the vascular system responds to stress. Anchoring its terminal rings at both surfaces, astaxanthin bridges the cellular membrane, acting as a microscopic strut that stabilizes architecture. When oxygen species attack, its carbon chain acts as a biological lightning rod, shuttling high-energy electrons outward to be neutralized by vitamin C. This mechanism is crucial for red blood cells. Packed with fatty acids and oxygen, they are highly susceptible to lipid peroxidation, which stiffens the cell. Clinical trials demonstrate that when astaxanthin protects these membranes from oxidation, the cells maintain their flexibility. In a controlled study, just 10 days of supplementation resulted in significantly faster whole blood transit times. Preventing microscopic membrane oxidation yields a macroscopic hemorheological benefit. By preserving mechanical flexibility, astaxanthin lowers overall vascular resistance, allowing blood to flow more efficiently. Inside the arterial lumen lies the trigger of atherosclerosis. When a highly reactive peroxynitrate radical strikes a native LDL particle, it mutates the lipid. Astaxanthin neutralizes incoming peroxynitrate radicals, transforming into 15-nitroastaxanthin before they damage the LDL. This intervention significantly delays LDL oxidation timelines. A 14.4 milligram dosage group showed a prolonged baseline, delaying oxidation by over 40% compared to the control. If LDL does become oxidized, it triggers a severe downstream pathological cascade, begin with the endothelial cells lining the blood vessel. A primary target in this cascade is the LOX1 receptor, a specialized protein sitting on the surface of the endothelium that binds specifically with these damaged lipids. Oxidized LDL binds directly to this LOX1 receptor, a signal that usually causes the cell to shut down endothelial nitric oxide synthase, or eNOS. However, as we see here, native unoxidized LDL cleanly bypasses the LOX1 receptor, leaving eNOS to function normally. Because astaxanthin prevents initial lipid oxidation, the LOX1 receptor remains unactivated, protecting the cell's ability to keep eNOS active. By limiting the triggers for eNOS inactivation, astaxanthin supports the sustained production of nitric oxide. This gas is required to maintain healthy vascular tone and allow for proper vasodilation. When oxidized LDL does manage to accumulate in the arterial wall, the vascular tissue launches an intense immune response. The immune system recognizes oxidized LDL as a threat, deploying chemical signals that recruit circulating monocytes straight from the bloodstream into the arterial tissue. Macrophages ingest the oxidized LDL, normally triggering the I-kappa B kinase complex to release NF-kappa B into the nucleus, commanding severe pro-inflammatory cytokine expression. Astaxanthin intercepts this by binding to the kinase complex, keeping the NF-kappa B inflammatory switch trapped and inactive in the cytoplasm. This intervention downregulates the genetic output of the cell. The production of interleukin-1 beta, tumor necrosis factor alpha, and the tissue remodeling matrix metalloproteinases drop significantly. The most critical genetic suppression for stopping cardiovascular disease is the downregulation of the macrophage's scavenger receptors, specifically CD36 and SRA. Without these receptor mouths, the macrophage loses the mechanical ability to ingest the surrounding oxidized LDL. By transcriptionally deleting the CD36 receptor, astaxanthin starves the macrophage of lipids. This arrests lipid accumulation, successfully preventing the macrophage's transition into a dangerous foam cell. Halting foam cell creation addresses one side of the equation. To clear existing lipids, the body relies on reverse cholesterol transport. The ABCA1 transporter normally transfers cholesterol to a nascent HDL particle, ApoA1. Inflamed macrophages secrete myeloperoxidase, which destroys ApoA1 structure and breaks this transfer. Astaxanthin intervenes, physically shielding ApoA1 from oxidative degradation, successfully restoring functional cholesterol efflux back to the liver. This cellular restoration translates directly to clinical lipid profiles in patients with moderate hyperlipidemia. After 12 weeks of astaxanthin supplementation, results showed up to a 25% drop in circulating triglycerides and a 15% increase in HDL. By stopping plaque formation at the receptor level and protecting HDL architecture, astaxanthin actively restores the body's innate biological mechanism for clearing existing arterial cholesterol. When we zoom out from the cellular level to the entire human vascular network, the complete clinical picture emerges. Astaxanthin's efficacy is driven largely by its precise geometric fit within the cell membrane. This structural alignment allows it to stabilize the bilayer while simultaneously acting on both the internal and external environments of the cell. First, it acts as a physical stabilizer and electron lightning rod, protecting erythrocytes and delaying the oxidation of native LDL. Second, it provides hard transcriptional inhibition of the I-kappa B kinase complex, trapping NF-kappa B and helping to prevent macrophages from converting into foam cells. Third, it biochemically preserves the architecture of ApoI1, re-enabling the reverse cholesterol transport pathway to clear existing lipid deposits. These microscopic mechanical actions correspond with measured clinical outcomes: reductions in oxidative biomarkers like malondialdehyde, lower systolic blood pressure, increased serum adiponectin, and significant drops in circulating triglycerides. The macro-level hemodynamic benefits of astaxanthin are the direct, measurable readouts of a precise dual-node transmembrane intervention. By intercepting the oxidative and inflammatory feedback loop at the source, it alters the biological progression of cardiovascular disease at the structural level.